Hello, everybody. This is Nathan Rayna again, and this part of our AR Cure workshop is talking about BioBrick assembly. So we've talked quite a bit, and you'll talk quite a bit about how to put these things together. But now we actually got to think about how we want to do BioBrick assembly and sort of the functions through BioBrick assembly. And so just sort of as a general guide, just some ideas to think about. One, go ahead and watch the alumni videos. I have several faculty who have done AR Cure over the years, have done a 5 to 10 minute video, just sort of talking about what it's like to do synthetic biology and how they use it at their institution. One of our big things is how I use it at my school may not be how you use it at your school. And so we try to bring a lot of alumni to talk about their stuff. The other thing is go to the IGM website and look for a team wiki and just start going through some wikis and find some cool things that are interesting. This is a project that we have students do the first week of lab, and I want you guys to go and look and see what's going on. It's important to me to have my students go to the IGM website multiple times, over and over and over again. And also, before you can begin this sort of series of work, is that you need to print out the Student 3A assembly slides. Those slides will be posted in the AR Cure Dropbox folder. And you really just need to print one slide per page. And you need to do the last slides with the plasma images. And so if you watch the video, go back and print them out and then watch the video again might be the best way to do that. And you only just need the plasmids and the images, not all the other slides. And you'll also need some scissors and tapes, and as we like to call them, restriction endonucleases and ligase. And so just sort of as a general background of where we're at in the class. The first week of class, students come in, I go over the syllabus and the project goals. And we do the IGM wiki review. So I put them in groups, I let them peruse through all the different wikis that IGM has. So it's really cool as every IGM team has to create a wiki. This wiki has all kinds of detailed information about their project. And so this is a really good way to see what's going on in synthetic biology. I don't tell students how to get there. I don't tell students much more than go to the IGM and look for wikis. There are a lot of different ways that they can Google, search, and find these, and I want them to come up with their own ways for this. Also in the first week, I give them an assignment that they take home and to start looking up basic BioBrick information. So I have an Excel file that has different parts of it filled out related to the BioBricks, their part number, their size, these types of information, and they have to fill that out, look it up on the IGM repository of standardized parts, and then come back and use those things. I also have them look up cut sites for various enzymes that we're going to use, and so I give them the list of enzymes for that. When I come into the second week of lab, this is really where we begin our work. Notice we're really actually working in the classroom at this point. We still have not learned any real lab skills. However, this is really important for them to start designing and creating their project, right? We're thinking like a scientist, and that's the first thing you have to do when you're doing these projects is design and understand your project. So today's lab, the second week of lab, we spend most of the time learning to use the list of standardized parts and how to interpret all the different information on their website. And we spend a huge amount of time going through the cut and paste cloning paper model experiment. This was an experiment that was given to me over 10 years ago. It has worked. I've done very little changes to it. You're welcome to steal it, use it for your own, just like I did. And then at the last part of the second week of lab is where they begin to discuss individual class projects. So they start thinking about what do other people do? What have other students done? What do I want to do? And we start making lists of what parts you might need and where you would look for these. So we spend a lot of time, me walking around working with these groups. And again, they're working in groups of 2 to 3, depending on what's going on. I like 3 better than 2. It's just easier for me to do it. When I got 2 labs of 20 students in each lab, groups of 3 makes it a little bit more manageable for me. So one of the things that I have the students do is go and look up the 4 restriction endonucleases that we use exclusively in the synthetic biology lab. It's EcoR1, PST1, XBAL1, and SBE1. These are the enzymes that you will need to order for your lab. I have students go and find the restriction cut sites and the recognition sequences for each one of these. And again, I don't tell them where to go. I just say, go find it. NEB has a really good website for it. Google, you can Google it and it'll almost show you up immediately what these are. iGEM also has. Just sort of as a note, when you're ordering from NEB, they're the company I really like to order from. They now have high fidelity enzymes, and the high fidelity enzymes do not work with this project. We have to do heat denaturation later on, and the high fidelity enzymes are not heat sensitive. And so do not buy those. And just sort of a general side note, they're awesome, but just don't buy them for this project. All right, so we've been talking forever about BioBricks. What is a BioBrick? And so here's the first introduction to really a BioBrick. It's essentially a plasmid, right? Everyone has seen plasmids. You guys have all done cloning with plasmids. But there's a few things to think about. I show my students this exact same slide, and we have this exact same conversation. All BioBricks, all plasmids, the plasmid portion of these are the same for every BioBrick that we're using. They're going to have an EcoR1 cut site. They're going to have an XBA1 cut site. And we'll call this upstream, right? Or they'll have a BioBrick in the middle. What makes every one of these different is the BioBrick that's inside of here. So when you get the iGEM kits, each well has a different BioBrick. And what that means is they're all in the same plasmid, essentially in the same plasmid. We'll talk a little bit about some variations. But what's different is the part that's in the middle. And so I always refer to this as the part. Downstream of the part is an SBE1 cut site and a PST1 cut site. They'll all have an origin of replication. However, something else to pay attention to is the antibiotic resistance. We'll see later on there are 4 main antibiotic resistances that your plasma could be in, and it's important that we go and look up what antibiotic resistance our part is in. And you can find all that in the iGEM website. Most of them are in chlorophenicol resistance plasmids when they come from iGEM themselves. It's also important to remind your students later on when they're doing cloning that they need to record what antibiotic resistance their plasma is in, because they're going to be moving things around throughout the course of the time of their class. And if you're not keeping up with what antibiotic resistance, you're going to run into problems. Trust me, I can do this. I may have said this before, but I know what not to do because I have done almost every boneheaded thing there is to do in BioBrick assembly and synthetic biology. All right, so what you need to do is you print it out. The first 2 plasmid slides just say Part A and Part B. What I'm going to show you now is a basic way how to put Part A into Part B. With the caveat, this is the way we used to do it, and it's not the way you will do it in the lab. I tell students the exact same thing. They stare at me blankly, but in my hands, I have found going this route helps them understand the next step, because the next step is too big a leap, right? So now we're going into the shallow end, we're easing into the deep end. However, we don't want to just jump directly into the deep end. I also make my students look up their parts. And so if you're looking at the plasmid that I have, there's a plasmid that we've made with PowerPoint. It's got all the recognition sequences. I make students first go find the recognition sequence, draw the little line, and show where they're going to cut for the different enzymes. And so first example, here's EcoR1. Here you can find the sequence. You can see how it's going to cut. But remember, we want to put Part A into Part B. And so I want to cut B completely out, and I want to attach it right in between these 2 parts. So really, we need to move down here and look at these enzyme cut sites. What you need to do on your part, for Part A, you need to find the cut site for SBE1 and the cut site for PST1. The first thing I want you to do, the first thing I have students do, is find that sequence and draw how it's going to cut with appropriate sticky ends. Once they do that, I give them scissors, which I call restriction endonucleases, and I give them tape, which I call ligase. For the rest of the class period, I only refer to it as ligase and restriction endonucleases. Anytime they call it scissors, I stare at them blankly and just look at them until they call it the right thing. Yeah, it annoys them, but you know, what can I do? It's the little things, right, that counts. So for your part A, I want you to find your cut sites for SBE1, your cut sites for PST1, and I want you to go ahead and cut them out, right? When you cut them out, you'll remove a little bitty insert that's between these two parts, but that's okay. On your part B, you're going to completely remove it from the plasmid. And so I want you to cut it on the XBA1 site, upstream of our part. And since we need to remove it, we got to identify a cut site downstream. And in this case, I want you to use PST1. So pause the recording, take a moment, and start cutting your parts and see what happens. What you should have after you've done these cuts is have something that looks like this. Now, I removed the other plasma digitally. If we were doing this in real life, doing this way, we'd have to run it through a gel. We'd have to do gel purification. We'd have to do all kinds of stuff to remove these parts, and that's why we no longer do it. That adds another 2 to 3 weeks to this process. With the new process that we'll show here late in a minute, it'll go much faster and much easier, and you can do it in almost one class period. So you have your parts, you have part B. If you look here, with my XBA site cut, you should have the right sticky ends. I have a part here with my PST1 sites with the appropriate sticky ends. And in part A, I have an SBE1 site with a PST1 also with the appropriate sticky ends. Now, I always call this part A is tethered to the plasmid, right? It's still attached to the plasmid. And our part is going to move and clone back into our stop. You have just now cloned your first BioBricks. Now I have part A cloned with part B. I don't call it anything in class. I literally just call it part A, part B at this point, because later on we start talking about orders of how we put genes together. We start talking about why a ribosome binding sequence has to be downstream of a promoter. We start talking about how we order these things, right? Because we're always going back to what we're talking about in the class, and we're trying to apply the big picture stuff. But right now, I just want them to focus on the cloning and the thought process. Now, one of the things I want you to start looking at is this middle spot. Go ahead, take a moment, and look at that spot, right? That's where our XBA and our PST-E1 sites overlapped. And look at that sequence and tell me, do you think that is the XBA site? Look at the sequence and see if it's an XBA site. And look at the sequence and see if it's an SBE1 site, right? Is that a recognition sequence for any one of those two enzymes? Do you see any issues? Can we cut that? What happens to those? All right? Think about all these types of deals, right? I stop at this point and I have students start trying to draw observations. What do we see? What are our problems? What are our issues? And this is where they go, well, what happens to this plasma chunk? What happens to this little chunk, right? I want them to start seeing problems. From the very first day of lab, they're starting to see and think about what are potential issues with this. The number one issue they come up with is this middle spot right here. Now, I already told you to go look at those sequences, and what you should have realized is that this sequence right here is not an XBA1 site, it's not an SBE1 site. The nucleotides are one off for each, thus the enzyme will not recognize or cut either one of these, and we call it a mixed site. Here's the other cool thing. We have the same cloning sites upstream and downstream as we did from the beginning. This is what makes this assembly really cool, is that no matter how once we put these together, once we figure out the pattern, we realize that we have our cut sites back together. Another observation that students finally realize is that the XBA sticky end and the SBE1 sticky ends are complementary to each other. different recognition sites, but complementary sticky ends. And when they clone together, we make a mixed site. And as far as we are concerned, as far as our students are concerned, this is permanent. So we go through the registry, we'll see composite parts where we've put part A to part B, and it's in the registry and we can use it. So we don't have to start with a bunch of small parts. We can start with bigger projects that have already been made. But those are permanently bound together as far as we are concerned. Now, there's other ways to do this. A lot of the iGEM teams will cut these apart, but for us and for our students, we consider these things permanent. So now you've made your parts. We have part A, we have part A, B, and we put them together. We have antibiotic resistance. You would put them into bacteria cells. You grow them up on one of your antibiotics, and we're ready to roll. But as I said, this is not what we do in the classroom. This is not what we call 3 antibiotic assembly. Students will do 3 antibiotic assembly in the classroom. And so now, after students spend about 30 to 40 minutes doing this part, we move to 3A assembly. I go back and forth. Do I want to give them the rules or do I want them to look up the rules? But the rules here are pretty simple. You're going to do 3 separate digestions. So you'll have 3 separate test tubes. And each test tube is going to be a different double digestion. One is going to be a linear backbone. We'll show you how to make linear backbone. You can also get linear backbone from plasmid, from iGEM. But really, it's a PCR version of the plasmid that doesn't have some of the middle inserts, and so it becomes a stuffer to put our parts in. We have our upstream part. And our upstream part, we're going to digest that with EcoR1 and SBE1. We have our downstream part, and we're going to digest it with XBA1 and PST1. And I think I forgot to mention our linear backbone is going to be digested with EcoR1 and PST1. And what we're going to do is we're going to digest all 3 of these separate tubes in their specific enzyme combinations. And after we digest them, we're going to heat and activate the enzymes, and then we're going to mix all 3 together. We're going to add ligase. I like T4 DNA ligase. What works the best in my hands is the NEB ligase. We do not buy the quick ligase. We do not buy the pre-mixed ligase. It works better for us if you buy just the ligase that comes with the buffer and do the mixes on your own. The quick ready mix does not really work well for this situation. The other thing you need to think about is your linear backbone needs to be a different antibiotic than your upstream and your downstream plasmids. So if I have a part A is in a chloramphenicol resistance, part B is in a kanamycin resistance, then my linear backbone needs to be in a tetracycline resistance. Because when we mix all these together and we ligate them, we're going to put them into our competent cells, and then we're going to use our antibiotic resistance that is in our linear backbone to select. So here's what I want you to do now. I want you to start going through the plasmids and see if you can design how you would put these together. See if you can cut and paste. A lot of times in the class, I don't give students this slide yet. I have them try to think through what combination of cuts they're going to do to put everything together. And so I let them work through this through a couple of weeks. But in the virtual standpoint, I wanted to show you this first. And so you can get an idea of the plasmids that we have. Here's a linear backbone. It's a kanamycin resistance. I have the sequences, and then I have the enzymes across the top for the students. And the students have to cut them out. They're all weird, you know. It's kind of interesting. They're all science students, and they're not art-minded, and they imagination sometimes gets to them, and so you have to spend some time cutting and pasting. They go through this, and we do this quite a bit. And so what I'd like for you to do is to print these out and start trying to see if you can put your parts together. So I have some parts that show the plasmid or the promoter, some parts that show ribosome binding sequences, and then I have green fluorescent protein. And start trying to cut and paste and design how you would do these clonings. First, put your promoter with your ribosome binding sequence and choose appropriate linear backbone. Then, once you put those together, take that new composite part and see if you can put it with green fluorescent protein and put it into its appropriate linear backbone. Students always want to cut steps here. People always want to cut steps because we're using paper and plasmids. We're not doing that. You need to think through how this would work in a tube. We will work as a group. One of the first things we'll do in the first AR Career Workshop virtual meeting is actually discuss this, work through groups, and try to work through how we do the 3 antibiotic assembly. This is the hardest part for students to get. Once they get this, they are ready to go, and the next part is designing their projects. Thank you. Have a good day.